So hello everyone and welcome to the ELSC seminar series again. Today we are hosting Josh Goldberg, an Associate Professor of Medical Neurobiology at the Faculty of Medicine here at the Hebrew University. Josh studied physics and cognitive studies as an undergraduate here at the Hebrew University, and he did his PhD in the ICNC with Chagai Bergman and Chaim Sampolinsky. He then did his postdoctoral training with Charlie Wilson at the University of Texas in cellular physiology of the basal ganglia. He then worked in the Israeli medical device industry for 3 years in development of active implantable system for vagal nerve stimulation to treat congestive heart failure. In 2009, he returned to the US as a research assistant professor at the Northwestern University in the lab of Jim Soumier, where he continued his work on cellular physiology of basal ganglia and related movement disorders. He started his own lab at the Department of Medical Neurobiology in 2013. His work focused on cellular neurophysiology of neurodegenerative diseases, mostly Parkinson disease. In recent years, he's interested in the pathophysiology of prodromal Parkinson disease, which he will probably talk about today. So before we start, as usual, we encourage questions during the talk. So feel free to unmute yourself and ask questions. And of course, there's going to be time for a question at the end of the talk. And without further ado, Josh, the stage is all yours.
I thank you, Didi. Thanks for inviting me to participate in your fabulous seminar this year. So I was a little bit worried, as I told you earlier this week, that you guys will be coming back from a retreat and nobody will want to hear another talk, but I understood that you had to retreat from your retreat because of the blazing fires. So maybe people are more interested to hear some more science. So I'm going to talk about the pathophysiology of prodromal Parkinson's disease. Let's see if I can move forward here. So as you know, Parkinson's disease is the second most prevalent neurodegenerative disease. It will affect 10 million people by the end of this decade worldwide. The main symptoms are bradykinesia, slowness of movement, akinesia, rigidity, difficulty to initiate movement, tremor, and postural instability. These symptoms are the motor symptoms of Parkinson's disease, are associated with the loss of dopamine cells in the substantia nigra pars compacta in the midbrain. However, we now know that Parkinson's patients suffer from a variety of non-motor symptoms. Some of those are listed here: dysautonomia and gastrointestinal dysmotility, things like constipation, dysphagia, difficult swallowing. There are sleep disturbances with a special group of patients suffering from REM sleep behavior disorder. These patients who are diagnosed with a sleep disturbance convert, 40 to 70% of them convert to Parkinsonism within a decade or so. So that's a group that's really prodromal. I'll mention that word again. People suffer from anxiety and depression, loss of smell, and even chronic pain. Now, in retrospect, patients, once they're diagnosed with Parkinsonism, can look back and say, we've had these other non-motor symptoms for a decade, 2 decades, with constipation being one of the earliest ones. And in fact, some of the clinicians, Nir Giladi from Tel Aviv University, actually say that there are various forms of these prodromal symptoms. So prodromal symptom is a symptom, by definition, that occurs before the actual diagnosis. The diagnosis, according to the motor symptoms, but these are prodromal symptoms that predate the disease. And we basically don't really know what is the mechanism that leads to these symptoms. However, what we do know is that in addition to the loss of dopamine cells, which is famously associated with Parkinson's disease, there's another pathology that's associated with Parkinsonism that was discovered by Dr. Friedrich Louis Levy over a century ago. And he described that in the postmortem brains of Parkinson's patients, he found these intercellular cytoplasmic inclusions where the main, we now know years later that the main protein, one of the main components of these Lewy bodies is a-Synuclein. And these are found not only associated with dopamine cells in the midbrain, but other places, and I'll expand that in a minute. And around the same time, it was discovered, many years after Dr. Levy, that the main component of these Lewy bodies is alpha-synuclein around the same time was also discovered that people that have families that have genetic mutations in the gene for alpha-synuclein also develop Parkinsonism, and therefore this gene has, in certain cases, definitely a causal effect in Parkinsonism. Now, this finding was elaborated by Heiko Braak, who basically discovered, looking at series of postmortem brains, that he found some brains that had these Lewy bodies in very caudal regions, like the dorsal motoneurons of the vagus, I'll be talking about extensively today. And he finds brains with the Lewy pathologies in those regions and nowhere else. And then on a later stage, he finds it in the dorsal motoneurons of the vagus and in the locus ceruleus. And then later on, he finds it in those two stations. And then also, he finds the Lewy bodies in the dopamine cells in the midbrain, and so on and so forth. And he described this sort of caudal progression of this pathology. And based on this, he defined 6 stages of Parkinson's disease, with stage 1 and stage 2 being preclinical, appearing before patients have symptoms. So only when these Lewy pathologies appear on the brainstem, and then you begin to have brain loss, that's when you begin to have Parkinson's disease. But obviously, there's this pathology that predates the disease, and that sort of parallels the fact that we have these non-motor symptoms that predate the diagnosis. So, many people think that clearly there must be, it makes a lot of sense that there's a connection between these things. So, for instance, maybe when the Lewy pathologies are in a very caudal region, like the dorsal motoneurons of the vagus, we know that region innervates the GI tract, and therefore that'll somehow associated with the GI dysmotility. And then maybe when it moves up to the locus ceruleus, maybe that has to do with mood or sleep, and so on and so forth. And Raphe Nucleus anxiety, so that the progression of the appearance of these prodromal symptoms actually have to do with this pathology. And that's a very reasonable supposition, but I think most people in the field, most pathologists think, if there's Lewy bodies, it basically is synonymous with cell death. So if there's pathology, it kills cells in these regions, and that's what causes these symptoms. You lose cells, vagal motoneurons, and that's what causes symptoms. That's definitely partially true, but the question is it really necessary for there to be actual cell death? Could it be that the fact that there's these Lewy bodies or even alpha-synuclein in these regions is enough to cause a chain of events to drive these prodromal symptoms, irrespective, regardless of cell death or prior to cell death. So in order to look at this question, we used the following model, which we borrowed from De Monti and Bonn, where we take an adult mouse, and we transfect it by injecting adeno-associated viruses harboring either a human mutated form of alpha-synuclein, the A53T, or not harboring anything, an empty vector, and we inject it into the cervical vagus of the mouse. And then, several weeks later, we have expression of the product, the protein, in the vagus nerve and in the brainstem. And that's what we see 6 weeks later. So as you know, of course, the vagus has efferent fibers and afferent fibers. So the afferent fibers terminate in areas like the area postrema, the nucleus of the solitary tract. And here you actually can see in this image that the protein product really is in sort of in terminal fields. However, there are also efferents that include the motoneurons in the dorsal motoneurons of the vagus. And here we can see that we find accumulation of alpha-synuclein within the cell bodies of these motoneurons.
Josh?
Yeah.
Can't see your cursor.
Ah, okay. Can I do that? I just write like this, right? Okay, so again, we have in the terminal fields, we see the these the proteins sort of in terminal fibers, and here within the dorsal motoneurons of the vagus, we see cell bodies that have accumulation of alpha-synuclein within the cell bodies. Okay, so first of all, does this generate a phenotype? Is this, the idea is that it's a snapshot of a very early time point. We have, this alpha-synuclein expressing in motoneurons. 6 weeks later, it doesn't seem to be anywhere else at this point. Does it have a phenotype that affects GI motility? And sure enough, the first thing we notice is that we have an increase in dry stool mass. And in order to characterize this a little bit more carefully, we actually measure the time of passage of food, through the animal's GI tract by gavaging it with tainted food and counting how long it takes. And we saw that definitely not 3 weeks, but 6 weeks after this transfection, there was a slowing of motility, and it took more time, for the food to pass through the animals. So we definitely have a phenotype in the mice transfected with alpha-synuclein, but now with the empty vector. So the first question is, maybe this is affecting the GI tract directly. So when we transfect here inside the vagus, we're transfecting fibers that are going to the GI tract. Maybe there's a direct effect there. So first of all, the human literature says you do find the protein around the plexuses of the enteric nervous system, but actually, people who looked at this in humans found that it seems to be within the terminals of the efferents. It doesn't seem to go into the GI tract, and this is actually was looked at more carefully by De Monte, who developed this in mice. At least in this model, it's definitely true that the protein reaches the efferents here on the GI tract, but it doesn't go into the internal enteric nervous system. But nevertheless, we wanted to look physiologically to see whether there was some kind of effect. So we used this classic preparation, an organ bath, right, where it's filled with physiological solution, and there's a little piece of ileum here from the mice. So it's fastened to a strain gauge that can measure the force. And if you look at this closely, you'll see that there are contractions. There are autonomous contractions. The GI tract has its own pacemakers, and it does its own contractions, and this happens spontaneously. We can measure those changes in force. We get this polymorphic oscillation in force, and when we look at like the spectral analysis of this and sort of try to extract the fundamental frequency, we actually don't see a difference. If anything, there might be a slight increase in the motility in these ex vivo pieces of GI tract of these mice. We also applied pilocarpine, which is a muscarinic agonist that directly affects the smooth muscle, and we can see that it causes a strengthening of the overall contraction. You can get a dose response curve, but we don't see a difference between the potency and efficacy. So it doesn't seem to be that there's a direct effect on the GI tract. Now this means that if we have a phenotype and the GI tract isn't affected, it's probably neurogenic, affecting the brain. So the first thing we wanted to do was look at the question of cell death. Maybe we have more cell death in the DMV, and therefore, that's why we have the vagal motoneurons release acetylcholine. It's part of the parasympathetic system. If we lose cells, there might be less of vagal tone, and therefore, vagal tone increases motility. Maybe we have cell death. So we looked at that, and we did a serological counting of cell loss, and indeed there is about a 20% cell loss due to the handling of this, this very sensitive nerve in the neck, stretching it. So there is cell loss, but it's equivalent in both treatments, both with the empty vector and the alpha-synuclein. So that cannot explain the divergence in the phenotype. And we also looked at another component, microglial activation. So neuroinflammation is, of course, famously tied with neurodegenerative diseases, also with Parkinson's disease. So at least we wanted to see whether this could account. Maybe we have more inflammation when the virus, the adenovirus, harbors the mutated gene. And again, here we do see some activation of microglia, but again, it looks like it's similar between both treatments. So we don't see none of these neuroinflammation or cell loss can't explain the phenotype. So then the next question we wanted to know is whether, okay, we've affected the vagal motoneurons with alpha-synuclein. Does that, if let's say they do affect the physiology of those cells, is that enough to slow motility? So for that we used dreads. So here I showed you, on the left I already showed you this result, but now we took a chat-pre mouse, which expresses pre-recombinase under the cholinesteryltransferase promoter, and we injected this with a pre-dependent GI dread, inhibitory dread. So in this case, we're only, when it's taken up in the vagus, it only goes to motoneurons because those are the only cholinergic neurons in the region. And then here, once we give the CNO, the ligand, so we get slowing. Okay, and again, if we just give the ligand on a non-injected mouse, it has no effect. So now we see that by slowing chemogenetically slowing the motor neurons in this region is sufficient to recapitulate the phenotype. Okay, so that's interesting. But the question is, do we have any evidence that the cells there actually slow down when they are transfected with alpha-synuclein relative to when they're transfected with antivectors? So for that, we injected, our postdoc, Wei-Hua Chu, you see his name here, transfected the mice, and we shipped them to my colleague Jochen Roper's lab at Frankfurt at the Goethe University. And then Wei-Hua traveled and spent cumulatively about half a year there before COVID-19. And he did juxtacellular labeling, so recording extracellularly in the dorsal motor nucleus of the vagus with juxtacellular labeling, so identifying which cells were recorded from. You can see that up here, and you can see where he recorded, and just looking at the firing rate, and sure enough, he discovered that there was a significant slowing down of the firing rate of these vagal motor neurons inside these mice in vivo. These are anesthetized mice. Now, interestingly, we had an interesting built-in control, because on our way down to the DMV, we passed through the nucleus of the solitary tract, which is more dorsal, and when he actually, so he had some recordings from those cells that fire an entirely different pattern faster, but here there was no effect. So there was no difference between the treatments between the basal firing rate of these nucleus of the solitary tract neurons, which are nearby, indicating this is really a selective effect for the cells, the motor neurons that took up this protein. Okay, so what could cause a changing of their firing rate in the presence of alpha-synuclein? So one thing could be some kind of synaptic network effect. But we do know that these cells are autonomous pacemakers. So even if you take them and look in a brain slice, and you take brain cells and you record for them acutely, and you put as many synaptic blockers as you want, you'll see that they're autonomous pacemakers. I also, in a previous study, I worked out the mechanism, so we know that this is the persistent sodium channel that's necessary and sufficient to drive this pacemaking. So these cells are pacemakers. So the simplest hypothesis is that somehow alpha-synuclein screws up, interferes with the pacemaking mechanism. And in order to look at that, we took brain slices from these mice and recorded their FI curves, their firing current relationships, and you can see those here. So sure enough, you see that the FI curve is sort of shifted to the right in the in the mice transfected with the A53T relative to those transfected with empty vector. So that indicates a lowering of excitability. You need more current to drive the firing. And you also can see that at zero current, there's a significant difference. But in order to quantify this more reliably, we did extracellular recording, so we're not interfering with the intracellular milieu with whole cell recording, and we quantify the slowing down. And again, lo and behold, we get a very similar result to the one we see in vivo, even in terms of not only qualitatively, but also quantitatively. The firing rate in vivo in anesthetized animals and in slices is roughly the same thing, and we get roughly the same effect. Okay? So now we've seen that the alpha-synuclein, the mutated alpha-synuclein taken up by these cells actually affects their pacemaking, slows down their pacemaking. But why? Why would it do this? Why? What is the mechanism? So, knowing, having worked on the biophysics of this neuron for over a decade now, we know pretty well what drives, what affects the pacemaking. One of the major important currents is this hyperpolarization activated and activating potassium current, the A-type current, that's very prominent. It's carried by Kv4 channels in these cells. So here in this study, we characterize, you can do the biophysics and extract activation. So we characterize, we know that this is an important current. And moreover, we know that the firing rate of these cells is super sensitive to changes in this current. So for instance, in a set of experiments where actually just simply from going from cell attached to whole cell, so breaking in, what happens is there's a rundown using whatever internals we use, there's a rundown of the A-type current. We measure it in time, and as that runs down, we see an increase in the firing rate. Here's a very dramatic example, extreme example, where the firing rate in cell attached is very low, and then when the A-type current gets messed up, it speeds up. We also did it in a more controlled way using this selective channel blocker called frixotoxin 2, which is selectively blocks Kv4 channels. And here too, we got an effect on the firing rate. So if you block this channel with frixotoxin, they speed up, right? I'm reminding you that a potassium current is sort of like a brake on pacemaking. So if you remove a potassium current, you expect it to speed up. So similarly, if there's some kind of an increase in the potassium current, we expect the firing to slow down. And then we'll talk about that in a minute. So the first thing we want to do is to determine whether this is actually the channel that's relevant to our phenotype. So what we did, we used this frixotoxin 2 and repeated the same set of experiments on the autonomous firing rate in slices. And when you pre-incubate in frixotoxin 2, you occlude the effect. So you no longer have a difference between the populations, indicating that it is this channel that's responsible for the effect. We also did this with submillimole concentration of 4-AP, which also is relatively selective, not as selective for A-type currents, and we get a similar result. So this indicates that we've sort of hit the problem. We figured out which is the channel that's relevant. Okay, so why again? So we have a candidate channel. There's a channelopathy that's causing these cells to slow down. Why is alpha-synuclein doing that? Okay, so for that we looked first of all at the biophysics of these. Again, this time it's done in these acutely transfected mice. So these are, as I remind you, adult regular C57 mice, but we've transfected some of them with or without. We don't see any gross difference in half activation, half inactivation. Kinetics looks pretty much the same. So we don't see an effect. However, interestingly, we did find that there was an increase in the current density. Okay, electrophysiologists, we measure the current and we normalize by the capacitance that we measure. And when you look at this, this is a proxy for getting a current, measuring the current density, and you see that we definitely get an increased current density in the transgenic mouse, which is consistent. If you have a higher current density, all of you guys who've done modeling and have solved a dynamical system describing a neuron, you know that it's the current densities that are responsible for charging the voltage. So if there's an elevated current density, that can explain a stronger effect for this potassium current, this larger break on pacemaking. And here it's quantified on the right by looking at the charge. We integrate underneath this curve. So we definitely see an indication of this. Now, the interesting thing is that if you just look at the total currents, you don't normalize by the capacitance, there is actually no difference between the groups. And that made us realize, if current isn't different, but we have a difference in the capacitance, something's happening to the morphology of the cell. And then we're looking back at the De Monte paper that first introduced this model. He actually mentioned there, he said that we see a slight shrinkage of cells that are expressing A53T. I know Ronit Sharon, I saw her in the crowd, she's also works on similar aspects of changes in morphology in alpha-synucleinopathies. So we went back and we looked at this, and we took animals that were transfected with alpha-synuclein. We looked at the cells that took it up versus the cells that didn't. So it's the same slice. We're doing morphological measurements of sort of the long axis of the soma, and we measure cells that either took up the alpha-synuclein, like here, ones that didn't, and we compared the distribution of those, long axes of the soma, and we definitely see this is a cumulative distribution. There's a leftward shift in the A53T treated mice. So this indicates that these cells are shrinking. Just as DeMonte had explained, we also did it, interestingly, in a subset of neurons that we had filled with biocytes, so we could sort of do a reconstruction using software like ImageJ of the surface area, an estimation of the surface area, in a handful of cells. We actually saw that you see this reduction in the somatic surface area. And I draw this next to the distribution of the capacitance that we measured from these cells. Again, a cell with a larger capacitance is larger, it has a larger soma. And interestingly, you see that both of these things behave the same way. Both these indications, the physiological measurement of capacitance as a proxy for cell size, shows that we have a reduction in cell size, and this morphological reconstruction. But the really striking thing was, and this really is one of my highlights, is Neuroscience 101, is that, for instance, you look at these two values, here, 22 around picofarad, and here about 2200 microns squared, and you normalize one by the other, you actually retrieve the exact specific capacitance of membranes, maybe except for humans, according to Idan, but the specific capacitance we learned from literature, actually. So these two different kinds of measurements, physiological and morphological, show that these cells have a smaller soma. Okay, so that basically what we've shown. Before what we've shown, I just want to say that this is very, very preliminary data for your eyes only. Charo Moratala from the Cajal Institute of Madrid has started looking at this in post-mortem tissue from very late stage Parkinson's patients. So this is not prodromal in any sense, but again, her initial measurements of her student, Monica's student, they show that there does seem to be this effect. Cells that have alpha-synuclein seem to be shrinking. So this is not, it seems like it's not it's not an artifact of our model, not necessarily an artifact of our model, because there's some indications. So that's just an interesting preliminary data not to be shared. So as an interim summary, what we've found is that a selective KV4 channelopathy, which is really basically shrinking, neurons shrinking, causing the effective surface density of KV4 channels to increase, that causes them to slow down their firing. And that, of course, in effect, when vagal motoneurons are firing less, they're releasing less, acetylcholine, less vagal tone. And that, as we saw with the GI dread experiment, is sufficient to explain this slowed phenotype. Now, of course, as you know, the vagus also carries cardiac vagal fibers, so fibers that go to the heart. And then, therefore, the prediction is that maybe the heart rate should be going up in these animals, because again, the parasympathetic system, it speeds up, rest and digest. It speeds up. The gastrointestinal system. So if there's a reduction in vagal tone, you see GAT motility going down. The opposite is true about heart rate. Parasympathetic tone to the heart slows it down. So if we have some kind of vagal neuropathy or loss of vagal tone, we should see the heart rate going up. And interestingly, a curious observation, when my postdoc Weiwa did these experiments in Frankfurt, he went ahead and induced anesthesia, and he very meticulous about measuring heart rate at specific intervals. And looking an hour after induction of anesthesia, he found that the heart rate of these mice were definitely higher. So they became tachycardic, which is consistent with this idea, another measurement. Now, why do I mention this? Because this is something that could, in principle, maybe be used to test on humans. So here I'm just showing an experiment, data from an experiment published by Dudi Al-Kadir here at Hadassah, where he actually looked at max VO2 in healthy controls versus mild Parkinson's patients. Mild meaning that their scores are not very severe. They haven't begun dopamine therapy. They've been diagnosed only within the 2 to 4 years, and he finds that when he asks these patients to either sit on a bicycle or start pedaling without any resistance, or then pedaling with 40 watts force, and then recovery, he finds that across the board there's a reduction in their heart rate. Sorry, the Parkinson's patients have an elevated heart rate. Now, of course, there are many reasons why this would happen. Your muscles are more rigid. There will be more energy expenditure, and it's definitely not this vagal tone. But the interesting thing in this finding is actually this slope. Okay, so we know, people who are cardiologists and do autonomy, estimation of the autonomic function. It's kind of agreed upon that the heart rate recovery from exercise is an indication of vagal tone. And here you see that there's this sort of the slowing down, the recovery is much slower in red versus to the healthy controls. So there is seems to be a component of heart rate recovery. Now this is something that can be done in principle on a patient. So somebody who might be worried that he has a bunch of symptoms that are prodromal could come and have an estimation of vagal neuropathy to see, and by the way, tests like this are done for diabetes patients. So the idea is that it could be possible these changes, these prodromal physiological changes that we've tried to characterize could also be read off simple, biomarkers, simple physiological biomarkers that could be looked at. Now, of course, I'm not claiming that we'll be able to diagnose Parkinson's disease by heart rate reductions, but this could be another biomarker used in addition to other biomarkers. Okay, so so summarizing, we see this effect of neuronal shrinkage causing slowing down of vagal motoneurons with this acute treatment of alpha-synuclein. But interestingly, a study that came out, 2014 by Jochen Roper, just before I began collaborating with him, he looked at firing rates in vivo and in vitro of dopamine neurons, the substantia nigra pars compacta, and sorry, there was a call there. And he discovered that actually this is a transgenic mouse. It's not acutely transfected. Transgenic mouse that overexpresses alpha-synuclein. He found that the firing rates go up spontaneously in these transgenic animals that overexpress alpha-synuclein. And moreover, he actually showed that this has to do with some sort of oxidative dysfunction of the same Kv4 channel. So dopamine cells, those of you who know the literature, famously also have a very big, A-type current. So what he did in this experiment was, what you see here are measurements of this inactivating current in 10-second intervals. And he's just showing sort of the amplitude of the current across time, but he has glutathione, which is an antioxidant, inside his recording pipette. So he's slowly introducing an antioxidant into these neurons while recording their A current. And what he discovered was that doing this actually causes their amplitude to increase. So there's a recovery of the A-type current in the dopamine cells using antioxidants. So, we have this sort of different phenotype. We see an indication of slowing down, but anyways, at the time, I said, okay, Jochen, let's look at the same animal. Let's look at the DMV neurons, do the same experiment. And then for another paper that I published, I believe Ronit Sharon is on this, and Mona Sorek also. He did the same experiment now on DMV neurons from essentially the same mice, and lo and behold, he got the exact opposite result. So if he if he washes in, dialyzes in glutathione into the wild type mouse, the DMV neurons, KV4 currents recover. But in the A53T, the neurons that overexpress A53T in this transgenic mouse, there's no effect. As if somehow these DMV neurons actually somehow respond by reducing their oxidative load, using some technique that dopamine cells obviously don't have. Now, when I was in my postdoc with Jim Surmeyer, Northwestern, we used a technique that he developed to study dopamine cells, trying to quantify mitochondrial oxidative stress using a fluorophore called Rho-GFP. It's a form of GFP that's redox sensitive. And anyways, using that, we were able to quantify the basal level of oxidative load in mitochondria of these neurons. So this is a way to measure mitochondrial oxidative load, at least in mitochondria. And the interesting thing in that paper was that by introducing isradipine, which is a plasma-lemmal CAV channel blocker, that actually totally the basal oxidative stress goes away. That is meaning that this elevated oxidation in the mitochondria is the result of calcium influx into these cells. And this is a general theme in Surmeyer's papers. You're invited to go look back at that. But we have this way of measuring it. So what we did, so the first question again is, if it indeed is, if we have this adaptive response, can we show that mitochondrial oxidative stress is reduced in the presence of alpha-synuclein? So what those experiments that Jochen did suggest that we should see a reduction in mitochondrial oxidative stress. And is it related to calcium currents? Because as we showed here in this paper 2012, the elevated oxidative stress is due specifically to calcium influx. So what if we, can we see a change in calcium currents? Because if there's a reduction in oxidative stress, it should somehow be related to calcium currents that drive it. Okay, so I showed you this result. We went ahead and received the construct, an AAV harboring the Rho-GFP, and this we injected into that transgenic mouse that overexpresses alpha-synuclein. And you can see that in the control, we actually were able to get very similar results to what we saw in 2012. However, in the A53T, indeed, there looks like a very strong reduction. So the transgenic mouse indeed has a lesser load, oxidative load in its mitochondria. And then to see whether this has to do with calcium currents, in these transgenic animals, we did a calcium imaging experiment where you patch a cell. This is wide field imaging with fluorophore, and we basically, injected either hyperpolarizing currents or depolarizing currents to change the firing rate of these neurons. So this is all done in whole cell, and we looked at the level of calcium as a function of firing rate. So that's what you see in this curve, and basically, if you normalize delta F over F by spike rate, you're basically getting some kind of proxy of a measure of how much calcium is coming with every action potential. And we found a significant reduction in the slope, indicating that there's less calcium influx. By the way, we repeated this experiment in the mice that we published this year. These data weren't published, where these are the acutely transfected, regular adult mice. And again, we saw the same thing. It seems like the amount of calcium coming in with spiking is reduced. So that now spiking calcium, of course, enters through high voltage activated channels. So if there's less calcium coming in, one of the simplest possibilities is there's a reduction in these these voltage activated calcium channels. So we went ahead and measured that both in the transgenic animal, and sure enough, that's what we see. This is a ramp. You give a ramp, you hold a cell in voltage ramp, you give a voltage ramp, and then you take the cobalt sensitive component, which gives you the calcium current, and sure enough, we see that there definitely is a reduction in the calcium currents in these transgenic mice in the DMV relative to the wild type. And that we also repeated in these acutely transfected mice, and here too, we see, not at 3 weeks, but at 6 weeks post-injection, we see a reduction in calcium currents. So it kind of ties in nicely. There's this vagal motoneurons somehow know how to respond to the alpha-synuclein in these transgenic mice and also in this acute transfection by downregulating their calcium currents. Now, just as an interesting point, when you normalize these measurements by capacitance and you look, then basically you don't see a difference anymore. So this sort of indicates, it's again, not very strong indication, but some kind of a hint that perhaps the cells know they're shrinking and they're able to change their molecular program to reprogram how much calcium currents they produce in order to normalize, but somehow, and it's very speculative, that fails with respect to potassium currents, and the potassium currents, because of the shrinkage of the cells, become larger, and as I showed you, that drives the slowing down. Maybe it's intentional, maybe it's because it's part, maybe it's helpful for the cell to slow down, because again, the slower the cell fires, less calcium comes in, and that's sort of protective. Okay, so I've shown you that acute inoculation of vagal motoneurons causes them to shrink, thereby selectively increasing the density of KV4 channels, selectively meaning that we didn't see the increase in the calcium currents, even though they are downregulated. Consequently, basal in vitro and in vivo firing rates are reduced. This effect is cell autonomous. Only cells expressing the protein shrink. And we also saw that neuroinflammation can explain this. And we also showed that GI peristalsis in this mouse is slowed 6 weeks after acute inoculation. We saw this heart rate effect as well. And I want to actually point out, I'm flashing here a paper that came out a few years ago, very interestingly, measuring gastrointestinal transit time in humans, in people with Parkinson's disease, and they found, using these magic pills, that measure that you swallow them and they spend time in your stomach and then in your small intestine and so on. And they found that there's a selective slowing of the proximal small intestine, okay? And that's super interesting because the vagus nerve innervates, there's this very strong gradient. The vagus nerve innervates the stomach and the small intestine, but it hardly innervates the large intestine. So the fact that they find in these Parkinson's patients slowing that selectively in the small intestine is really, really interesting in this respect. Okay, so our conclusion is reduction in vagal output may contribute to GI dysmotility in prodromal Parkinson's disease. To the best of our knowledge, this is the first demonstration of a causal chain of events from selective channelopathy in a distinct class of neurons to prodromal symptom of PD. And the neurons, at least in this model, are intact. I'm not saying that there is no neuronal loss in these regions. There is in humans, maybe to a lesser extent than in dopamine cells, but at least this effect doesn't require cell death. It doesn't necessarily, right? Preliminary postmortem data suggests that shrinkage of vagal motoneurons also occurs in PD patients, and we are considering, possible biomarkers with Dudia Kadil to use, to develop biomarkers looking at these physiological readouts that could give you an indication of some change in vagal tone in early Parkinson's disease, or maybe prodromally. In addition to slowing pacemaking observed in the adult onset model of alpha-synucleinopathy, downregulation of calcium channels reduces activity-dependent calcium influx in vagal motoneurons, both in the transgenic mice and in the adult onset model. Mitochondrial oxygen stress is reduced in the vagal motoneurons of the transgenic mice. In addition, in these mice, the KV4 channels exhibit oxidative dysfunction in the dopamine cells, but not in vagal motoneurons. And taken together, this, perhaps differential adaptations in calcium and potassium channels may explain why vagal motoneurons are more resilient than dopamine cells to Lewy pathologies. Okay, so, right, and then the final thing I wanted to talk about was the title of my talk, right? Is, maybe this is a new era of pathophysiology of prodromal disease. So when I was a graduate student, I trained with Chagai, who's one of the champions of defining, the physiological pathophysiology of end-stage Parkinson's disease. MPT primates, you lose all your dopamine, it's an end-stage stage, and we have a very good understanding of a very broad characterization of changes in physiology that occur in late-stage Parkinsonism. That's also, we also see in the clinic through the surgery, we can see changes in firing patterns of the basal ganglia. But I think this is sort of a new era of looking at the pathophysiology of the very other end of Parkinson's, the very early state. And in our lab and with other collaborators, we're trying to follow up on this idea. So Gilberto Fisoni here is in the crowd, he's one of our collaborators on this grant with Professor Martella, whom I mentioned, and Gilly Silverberg and others, looking at the prodromal symptom of anxiety and depression, and basically trying to look at the same kind of things, look at Raphe Nucleus, look at dopamine cells in the Raphe Nucleus. This, by the way, this image belongs to Gilly. I should have given it. This is Gilly Silverberg's. I forgot the credit. This is his image from his lab. But here's a measurement. We'll be getting to calcium currents also in the dorsal motor nerves of the vagus. Sorry, in the DRN, the dorsal Raphe nucleus. So that's our target for studying anxiety and depression in this grant. And I'm also submitted recently a grant with Alex Binshtek about pain, which there's some indication that chronic pain might be a prodromal. So we're also looking perhaps at that. So just to mention the people who did the work. So Wei-Hua Chu was a postdoc, fabulous, outstanding, brilliant postdoc in the lab. He moved on to NYU, just as the corona began. He was there when things were already shutting down in Israel, but New York was still partying, and he was sort of felt like in zombie land for his first week there. He was sort of locking himself up in his new apartment, and people were still partying. Anyways, he's at NYU now. And he did, most of everything I showed you. Ruth Musgrove did the stereology and neuroinflammation experiments. And of course, I mentioned Jochen Roper, whose lab we did the experiments of the recording in vivo. David Al-Qadir, we're trying to work together in humans, and Charo Maratala gave us the preliminary data showing shrinkage in human patients. So thank you very much.
Thank you, Josh. We have time for questions. So Josh, maybe I can ask the first question. So you focused on one particular clinical symptom of Parkinson. So you think the same mechanism might play a role in other prodromal or later symptoms?
Right. So that's exactly the two last slides I flashed after my time was up. So I went through them too quickly. Is that exactly what I'm trying to do with various colleagues? Is to say, okay, we see this. We see it. We see a change, cell-selective changes, even a channelopathy you can describe. But vis-a-vis the Raphe nucleus, there are dopamine cells in the Raphe nucleus, and actually, Glenda Halliday showed. I forget already, a long time ago, she showed that there's cell loss in the Raphe, and there's a preferential cell loss of dopamine cells in the Raphe nucleus. And the Raphe nucleus, for obvious reasons, we have Iran here maybe in the crowd, but Iran is definitely the local expert on those cells. Yeah, I see his name here. So those are associated with many things, but also anxiety and depression. So we're trying to do the same kind of experiment. We have a student in the lab, Nadine Wattad, who's doing these calcium channel measurements in dopamine cells in the DRN, and then she's hopefully can continue to do a PhD with me, and she'll transfect that region with a-Synuclein and sort of try to see whether this idea of a specific channelopathies by selective transfection with a-Synuclein also can drive these other phenotypes of anxiety. Again, I mentioned Gilberto here in the crowd is of course a major force in that. He's an expert on that idea. So that's one thing. We're hoping to look with Dudi Al-Qadir at changes in heart rate, in mice, maybe even humans. And as I said with Alex, we just submitted a big grant to try to look at prodromal pain, because we also know that the pain modulatory system that modulates the sensation, not nociception in the spinal cord, includes some of these regions that are actually vulnerable to synucleinopathy in Parkinson's patients. So we're trying to see whether there's also maybe this chronic pain that not many, but some Parkinson's patients, their initial complaint is pain, and it's treated sometimes orthopedically, like frozen shoulder, but whatever. We know that chronic pain is in some cases could be prodromal. So Alex and I are hoping to look at that question as well. So yes, we're trying to generalize it to other prodromal symptoms, and using the same methodology.
Vital, I see that you raise your hand. Do you want to ask a question?
I'm asking about which vagus we use. We use the right vagus. That's important, at least in cardiac physiology. I mean, actually now there was a beautiful paper also about the GI tract. The vaguses on both sides don't exactly do the same thing. Heart physiology, we know that the right vagus innervates the SA node, and the left vagus innervates the AV node. And there was a beautiful paper last year showing that the vagal system also has the differential, left-right differential regarding the GI tract. So we worked systematically on the right vagus. We didn't look at the left vagus. I don't think it really, probably doesn't matter. I mean, I don't see why, unless you have a good hunch to explain. Let's see who else.
Is it okay to ask something?
Yes, of course.
I was wondering about the shrinkage. Do you know if the neurons fire less? The same neuron is sort of dying, so it's firing less, or is it some parts or some neurons are dead, or is it a gradual disorder, sort of not firing all the time, but sometimes?
So that's a legitimate question. I mean, we don't think, as far as we can do physiology, recording whole cells, cell attached, the cells don't look, worse off. And essentially, it could very well be that this is a process that's declining, towards cell death. But at least when we patch onto them and record from them, they look sort of normal as far as we can tell. We actually looked at other measures. I have a figure of other measures of their physiology: spike width, spike threshold, slow HP. All these measures, we don't see a difference. So I can't categorically say that these aren't dying cells, but it's not not something acute that's happening in the slide. I mean, the cells look roughly the same as the other ones, and we can get, usually when cells are really dead, it's hard even to patch them and do things like that. So it could be that in the long run those cells are dying, but we don't see, as I said, a major indication of cell death when we count it at the level at this time point. It doesn't seem to be.
You said it's firing less when the cells are shrinking. So I'm wondering if some of them are working some of the time, or
how regular it is. So I have in our I'd say it's as regular as we see. I don't think it's like, didn't change dramatically the firing time. They roughly, they're pretty tonic. I showed you an extracellular recording. They're pretty tonic. Yeah. It might be a little bit of a jitter, but it's not like they've, changed their firing pattern altogether as far as we can tell. No major changes. So, but the real important question is or point is that we don't actually know the mechanism of the shrinkage, and it's something. I think I'm talking to Onit Sharon in the crowd about it, and I think people understand cell physiology and all sorts of pathways, and that sense might have better hunches, and I would love to collaborate with him. I don't really know to say why alpha-synuclein is causing the cells to shrink. Onit, I think has a better hunch about that, and I'm hoping maybe to work with her about this at some point.
So if I may, so Josh, so actually, if I understand correctly, so you see a correlation between alpha-synuclein and the shrinkage, right? But it's not clear if it's causally related to the shrinkage, right? It might be a byproduct.
So what's causally related? I mean, what we see is that the shrinkage, we can also measure. We see its effect on the physiology. We see the capacitance of the cells are smaller.
Yes, that's the pathophysiology, but what's the mechanism between alpha-synuclein and the shrinkage? It's not clear, right?
Basically, I don't know. I don't know to say that. Ramit had a paper about the ramification, changes in axonal morphology in the stratum and using alpha-synuclein. And I think actually there's actually more surface area and become more ramified. I don't remember. I hope I'm not quoting it very imprecisely. But it's not the first. We're not the first to observe this effect on morphology. But here we've been able to tie it, we think, pretty reasonably well with its effect on the pathophysiology.
Any more questions? Okay, so I think we'll stop here. Thank you very much, Josh, for a very interesting talk. And see you everyone next week.